Tuberculosis is a devastating and deadly disease, against which present countermeasures are inadequate. It is one of the most widespread of the infectious diseases. Shockingly, 1/3 of the human population is already infected. The new EU member countries of Eastern and Central Europe are at an elevated risk due to ever-increasing infection rates and decreasing control of active and latent TB cases. Tuberculosis was once known as morbus hungaricus, as in the early years of the last century, Hungary had very high rates of infection. During the 1960s, a successful campaign led to a drastic reduction of active TB cases, and the rate of new infection was also driven down. However, due to fewer restrictions, increased international travel, and a lack of systematic screening, the number of TB cases is on the rise again. The same factors are at play worldwide, creating a situation which demands our attention. Previously successful drugs may have led to a degree of complacency, which is a liability under current circumstances. Soon after the introduction of the first anti-TB drug, streptomycin, in the last century, drug-resistant strains began to appear. At present, uncontrolled treatment and unsatisfactory compliance to therapy regimen are causing a rise in the incidence of multi-drug-resistant TB.
From Hungarian funding sources, a large multidisciplinary research project started this year. The project is aiming to find new drug candidates against tuberculosis. The members of the research team include the Eötvös University, the Institute of Enzymology of the Hungarian Academy of Science, and two private companies, Euratim and Ubichem Limited. Our project is focused collaboration between mathematicians, computer scientists, physicists, chemists, and molecular biologists. In what follows, we are trying to give an impression what we are doing, why it is a new and innovative approach to this old problem of mankind. At the Euratim Limited, mostly mathematicians work in the project. While it is not especially hard to imagine what biologists or chemists do in a health-related project, it is perhaps hard to imagine what mathematicians do here. Two main features of our work is the target identification and docking simulation.
Organisms have several thousands of enzymes and other proteins encoded in their genes. A bacterium can use several hundred different enzymes for its metabolic activity, for example growth and reproduction.
For the target identification, our goal is to kill the bacteria. In order to do this, we need to choose some processes in the bacterium and need to disturb these processes lethally for the bacterium.
In a cell, proteins pass molecules to each other in the same way soccer players pass the ball to each other while advancing downfield. If you consider the tuberculosis bacterial cell to be a football pitch, we can imagine the TB native proteins to be the players on the home side. Similarly, the drugs we seek to fight it with could be seen as a highly outnumbered guest team. If the guests block the single best player of the home side, the latter can quickly adapt by replacing their star with a series of other team members. Whereas if the guests block several of the strategically most important players, they have a good chance to cut off a pass and end the offense before it has a chance to strike.
We use large graphs to identify the vital proteins in the tuberculosis bacterium.
Now I'm going to talk about how we can construct the metabolic interaction network of the tuberculosis bacterium. This metabolic interaction network consists of approximately 1,000 nodes that correspond to chemical reactions catalyzed by the enzymes of the bacterium. The nodes are connected with an edge if the corresponding reactions share at least one small molecule. This metabolic interaction network can be constructed using publicly available databases. Our goal will be to find a suitably small set of reactions in this metabolic interaction network, blocking of which is fatal for the bacterium. More precisely, we would like to choose between 30 to 50 target proteins with known three-dimensional structure, the existence of which is crucial if you want to use in silico docking algorithms. Finding the appropriate reactions in the metabolic interaction network translates into the task of finding important nodes of this graph.
I would like to emphasize that we aim to find not an important single protein of the bacterium, but rather protein pairs, protein triples or quadruplets to attack them simultaneously.
Theoretically, it is possible to predict the position of a small molecule in the binding cavities of a protein. In practice, though, it's quite a challenging task.
The traditional way of finding a drug against various bacteria is using various docking method to find small molecule which binds specifically to a protein which is vital for the bacteria and which not appear in the human cells.
It is well known that molecules accept the position with the lowest possible energy.
Euratim developed the frigate docking algorithm for fast and reliable docking of millions of small molecules to the surface of one protein in about a week. The computation used about 100-150 processors in parallel.
The molecule can move in several ways. It can translate, rotate in space, and twist around some of its bonds.
The problem of finding a minimal energy conformation is the higher dimensional equivalent of the problem of finding the deepest valleys of an alien planet via the use of short-sighted robotic probes. In this metaphor, a lander that is capable of measuring altitude and steepness once landed would be equivalent to the calculation of the energy belonging to a given conformation. As the most expensive operation of search algorithms is the calculation of energies, the fewer landers we need, the faster and more efficient the algorithm becomes. Scanning all of the conformations with high resolution would take even a very fast computer millions of years. The goal is to create an effective algorithm which can be run in a reasonable time frame. To this end, mathematicians have incorporated self-educating heuristics into the software which direct it to avoid regions of higher energy and focus more fruitfully on lower energy areas.
If we have two atoms interacting, then finding this energy minimum is like having a nice walk on the side of a mountain down to the closest valley. But when we have hundreds of protein atoms interacting with about 20 to 30 atoms of a small molecule, it's more like being lost in the middle of a jungle.
There is another approach which we also use this project, and it's based on the fact that many of the protein has no such a structure, but they natively unstructured proteins. These unstructured proteins play a vital role in protein network, so attacking this protein can kill the bacteria by inhibiting the network formation in the bacteria.
EuBIChem Research is one of the largest independent Hungarian pharmaceutical research and development organization. EuBIChem is doing or providing services for various size pharmaceutical companies, including small and virtual pharmaceutical companies, as well as large international pharmaceutical organizations. Our main function is to synthesize or make new pharmaceuticals for clinical trials for experimental purposes, as well as analytical work, structure identification of the synthesized compounds. As part of the consortium, EuBIChem's job is to synthesize novel candidates for the potential treatment of tuberculosis. And hopefully these potential candidates later on can be turned to a successful API for the treatment of tuberculosis.
Our colleagues identified new compounds which potentially active against Mycobacterium tuberculosis. Our task is to verify these findings by experimental manner.
Another way to verify the efficacy of the in silico identified drug candidates is to add these compounds directly to the bacterium.
Experiments with live Mycobacterium tuberculosis cultures require a highly safe laboratory environment. In this project, the Korenyi National Tuberculosis Center provides these facilities where the identified promising compounds can be tested in vivo on the infectious bacteria.
In this experiment, first we make a dilution, then we add the compounds in 10 different concentrations to Mycobacterium tuberculosis suspension in liquid media. Then we need to wait 4 weeks because this is a very slow-growing bacteria, and then we can determine the minimal inhibitory concentration. The next in vitro experiment is to measure the cytotoxicity or the cytostatic effect of the compounds.
Even in case of the destroyment of the bacteria of the Mycobacterium tuberculosis within the cells, we are there, we have the compound which are suitable for further consideration, and it is a good basis for developing a new drug. Further improvement could be made when we conjugate this potentially active and experimentally active drug with short peptide sequences. These peptide sequences could enhance the selectivity of the drug, by targeting the compound to macrophages which are infected.
First, we make the carrier peptides on solid phase using an automated peptide synthesizer.
Optimal conjugation methodology for the covalent attachment of the drug to the peptide. So first thing, to select the appropriate peptide sequence for the targeting for increasing specificity. The second thing is to develop methodology for the appropriate conjugation. In this respect, we need some sort of covalent bondage which cannot destroy the activity of the drug, but potentially within the cells capable for a cleavage, which means the drug will be liberated and could be active within the cells, within the macrophage which is infected by Microbacterium tuberculosis. The purified compounds are going then to be tested for verifying the chemical structure. That means we need to verify that the peptide is there, and we also need to have experimental evidences that the drug is there. For that, we are using, for example, mass spectrometry as a contemporary and most fast methodology for these purposes, but we are also using traditional, well-established techniques like amino acid analysis, which is telling us that the peptide is there, or spectroscopic experiments, for example, fluorescent spectroscopy, which is suitable when we have the drug with fluorescent properties.
Flow cytometry is an effective tool to investigate cellular uptake of our ligand conjugates by human cell lines like human macrophages. Fluorescent or fluorescently labeled compounds can be detected in the cells already at low concentrations. Binding to the cell surface also can be examined at low temperature or using fixed cells. Concentration dependence, kinetics, and temperature dependence of the uptake can be studied.
This is the Laboratory of Genome Metabolism and Repair that belongs to the Institute of Enzymology. The Institute is part of the Hungarian Research Academy, and therefore we are one of the academic participants of the Jedlik project. The main aim of the project is to fight Mycobacterium tuberculosis by innovative novel means, by identifying new protein targets within the bacterium and also making use of the knowledge gathered from networks. Now, what our contribution might be in such a project? What can we contribute from basic research? I find this is a major challenge, and a challenge that's also motivating for us. We have two main contributions, I would like to say, and one comes from the basic research knowledge that we gathered in the last decade. As a research laboratory, we have been focusing on the problem of DNA repair. And this problem actually faces a major biological paradox that can be phrased like that. We all know that DNA is responsible for storing genetic information in a stable way. However, DNA is not stable. From a chemical point of view, this is a highly reactive macromolecule that also exists in a highly reactive microenvironment within the cell. So actually it's like an unstable macromolecule within an unstable environment is responsible for stable information storage. Now, of course, this is not possible. There have been major solutions to this problem, and the solutions reside in the DNA repair mechanism. We are targeting the DNA repair mechanism in order to understand how these mechanisms restore the stability of genetic information. And since these mechanisms are essential for all cells, for bacterial cells as well, our aim is now to use the basic knowledge to fight Mycobacterium tuberculosis by targeting the DNA repair system of the bacterium. And on the other hand, we are also using a technological platform. The technological platform which we are using actually comes from structural biology. And everybody knows a major discovery of structural biology for which the Nobel Prize have also been awarded, that was the determination of the structure of DNA. We are now capable of determining macromolecular structure in such a way that we will know where each atom is located in the three-dimensional space. We are using this knowledge, and we hope that by this contribution, important novel drug candidates will be developed.
We deal with modeling of drug transport through cell membranes in the frame of TB Inter project in the Laboratory of Interfaces and Nanostructures of Chemical Institute. The drugs against Mycobacterium have to go through several interfaces in the human body, like macrophage membrane and also bacterial cell walls. We use a simple monolayer model of membrane to investigate the drug transport processes. The material of this monolayer is a combination of the main lipid components of the cell membranes. The lipid layer is prepared in a Langmuir balance. A Teflon trough is filled with water. The lipid solution is spread on the water surface. With help of two barriers, we can decrease the area so the molecules are much closer to each other, forming a close-packed ordered structure. When we have reached the compactness of the cell membrane, we stop the barrier. The degree of drug-lipid interaction might indicate the probability of